Welcome to our lecture on water quality assessment. Today, we are going to explore two of the most critical measurements in aquatic biology: biochemical oxygen demand, or BOD, and chemical oxygen demand, or COD. If you have ever wondered how scientists actually measure how polluted a river or lake is, you are in the right place. By the end of this session, you will understand exactly how we quantify invisible pollution and protect our aquatic ecosystems. To begin our journey, let us imagine you have a beautiful, clear fish tank, and you accidentally spill an entire container of fish food into the water. You might think the fish will just eat it, but what actually happens is that millions of invisible bacteria start feasting on the decaying food. Just like humans and fish, these bacteria need to breathe oxygen to survive and digest their massive meal. As they multiply, they consume almost all the oxygen dissolved in the water, leaving your poor fish gasping at the surface. This exact scenario happens in our rivers and lakes when sewage is dumped into them. Now that we understand the basic mechanism of oxygen depletion, let us look at how we measure it scientifically. Biochemical oxygen demand, commonly abbreviated as BOD, is defined as the amount of dissolved oxygen needed by aerobic biological organisms to break down organic material in a water sample. In standard laboratory practice, this test is conducted over a period of exactly 5 days at a constant temperature of 20 degrees Celsius. This is why you will often see it written as BOD5. By measuring the oxygen levels before and after this 5-day incubation, scientists can determine exactly how much oxygen the bacteria consumed. A high BOD value means the water is highly polluted. Understanding what BOD is naturally leads us to why it is so absolutely critical for environmental protection. Whenever a city treats its sewage or a factory releases wastewater, we must ensure that this water will not destroy the local river ecosystem. If wastewater with a high BOD is released into a stream, the natural bacteria will rapidly deplete the river's oxygen. This leads to massive fish kills and the creation of dead zones where no aquatic life can survive. Therefore, BOD serves as a universal warning system, telling us exactly how much damage a specific effluent will cause to the oxygen balance of a receiving water body. Since raw sewage would consume all the oxygen in a test bottle within hours, we must dilute it to calculate the BOD. This formula we just saw on screen relies on the difference in oxygen concentrations. Conceptually, it calculates the total oxygen consumed by the bacteria in the diluted mixture and then scales it up to find the demand of the pure wastewater. D1 represents the initial dissolved oxygen concentration of the diluted sample, measured in milligrams per liter. D2 is the final dissolved oxygen after the 5-day incubation. P is the decimal volumetric fraction, which is simply the volume of polluted water divided by the total volume. If the initial oxygen is very high and the final oxygen is very low, the numerator becomes large, resulting in a high BOD. Before we move on, let us see this with a concrete example. Suppose an environmental scientist collects 10 milliliters of highly polluted wastewater and dilutes it to a total volume of 300 milliliters. Because the wastewater is highly polluted, she cannot test it directly. First, we must calculate the dilution factor, which is the fraction of the actual wastewater in our total mixture. We divide the volume of the wastewater sample, 10 milliliters, by the total volume of the diluted mixture, 300 milliliters. This gives us a volumetric fraction P of 0.0333. This tells us the actual polluted water makes up only about 3.33% of the liquid in the test bottle. Now that we have our dilution fraction, we can complete the calculation. The scientist measures the initial dissolved oxygen and finds it to be 8.5 milligrams per liter. After keeping the bottle in a dark incubator for 5 days, she measures it again and finds it has dropped to 3.5. We substitute these known values into our main BOD equation. We subtract the final oxygen from the initial oxygen to find that 5.0 milligrams per liter were consumed. Dividing this consumed oxygen by our dilution fraction of 0.0333 scales the result back up. The final answer is 150 milligrams per liter, meaning dumping this raw wastewater directly into a river would instantly strip the water of all its oxygen. Now that we have successfully calculated BOD, we must address a major flaw in this method. The BOD test takes 5 whole days to get results, and bacteria are picky eaters. Imagine trying to clean up a toxic chemical spill. The bacteria in your test bottle might simply refuse to eat the chemicals because they are poisonous or non-biodegradable. You would look at your BOD test 5 days later, see that no oxygen was consumed, and falsely conclude the water is perfectly clean. To solve this problem, scientists needed a much more aggressive method that does not rely on living, breathing bacteria to slowly digest the waste. This need for a better method brings us to chemical oxygen demand, or COD. COD is a measurement of the total quantity of oxygen required to chemically oxidize all organic and inorganic compounds present in a water sample. Instead of relying on bacteria, we use powerful, harsh chemicals, typically potassium dichromate, to forcefully burn and break down absolutely everything in the water sample. This test is performed by boiling the sample in a highly acidic environment for about 2 hours. Because this brute force approach destroys both biodegradable waste and tough non-biodegradable pollutants, the COD value of a water sample is always higher than or equal to its BOD value. To understand how we quantify this chemical destruction, let us look at the COD formula. Conceptually, this calculation is based on a laboratory technique called titration, where we measure exactly how much of a strong chemical oxidizer was used up by the pollution. VB represents the volume of the titrant used for a blank sample of pure water, measured in milliliters. VS represents the volume of titrant used for the actual polluted water sample. N is the normality of the titrant chemical, which tells us its exact concentration. The number 8,000 is a constant conversion factor that translates chemical equivalents into milligrams of oxygen per liter. Finally, V is the volume of the original polluted water sample. If the polluted sample consumes a lot of the oxidizer, VS will be small, making the numerator large and indicating heavy pollution. Let us apply the COD formula to a real-world scenario. An environmental engineer takes a 50 milliliter sample of polluted water near an industrial discharge pipe. After the digestion process, he titrates the sample using a chemical with a normality of 0.1. The titration of the polluted sample requires 12 milliliters of titrant, so VS is 12. He also runs a blank test using pure distilled water, which requires 25 milliliters, so VB is 25. First, we find the difference between the titrant used for the blank and the sample. Subtracting 12 from 25 gives us 13. This difference represents the exact amount of the chemical oxidizer that was consumed entirely by the pollutants in the water. With our titrant difference calculated, we can now find the final chemical oxygen demand. We substitute our difference of 13, the normality of 0.1, and the constant 8,000 into the numerator. We multiply the normality by the constant to get 800, Then we multiply 13 by 800, which gives us 10,400. Finally, we divide this result by the original sample volume of 50 milliliters. The final calculation reveals a chemical oxygen demand of 208 milligrams per liter. This means it would take 208 milligrams of oxygen to chemically burn away all the pollutants in just 1 liter of this water. This captures both biodegradable waste and tough toxic chemicals. Now that we have calculated both values, let us look at this diagram to visualize their relationship. On the vertical axis, we have the oxygen demand, measured in milligrams per liter. The blue bar represents the biochemical oxygen demand, which only captures the waste that bacteria can naturally digest. The red bar represents the chemical oxygen demand for the exact same water sample. Notice how the COD bar is significantly taller. The gap between the top of the BOD bar and the top of the COD bar, indicated by the arrow, represents the non-biodegradable organics and toxic inorganic chemicals that bacteria cannot process. This visual perfectly illustrates why COD gives us a more comprehensive picture of total pollution. To wrap up our lecture, let us review the key takeaways. Biochemical oxygen demand tells us how much oxygen living bacteria will consume as they clean up organic waste over several days, simulating what naturally happens in a river. Chemical oxygen demand, on the other hand, uses harsh chemicals to instantly measure absolutely every pollutant in the water, whether bacteria can eat it or not. Because COD destroys everything, its value will always be higher than the BOD value for the exact same water sample. By comparing these two numbers, environmental scientists can figure out exactly what kind of pollution is in the water and design the perfect treatment plant to clean it up. You now have a solid understanding of how we assess water quality.